Assalamu alaikum class. Welcome again to the series of lecture on the extensions and modifications of Mendel's laws. In the previous two lectures on the extension and modifications of Mendel's laws, we have covered the phenomena of incomplete dominance, codominance, multiple alleles, lethal alleles, penetrance and expressivity, and epistasis. In this last series of lecture, we are going to study about pleiotropy, genetic heterogeneity, and phenocopy. Let's get started. First of all, we have the phenomena of pleiotropy. For Mendel's experiment, you might imagine that all the genes control a single characteristic and affects some harmless aspects of an organism's appearance, such as color, height, or shape. Those predictions are true for some genes, but definitely for not all of them. For example, in case of humans, there's a genetic disorder called Marfan syndrome, and it is caused by a mutation in one gene. Yet it affects many aspects of growth and development, including height, vision, and heart function. This is an example of pleiotropy, or one gene affecting multiple characteristics. When we mentioned about Mendel's experiment with purple flowers and with white flowered plants, we didn't discuss any other phenotype associated with the two flower colors. However, Mendel noticed that flower color was always correlated with two other features. First of all, the color of the seed coat. Coat is the covering of the seed, and the color of the axils, which is actually the junction where the leaves meet the stem. In plants with pure white flowers, the seed coat and the axils were colorless. In plants with purple flower, on the other hand, the seed coat were brown, gray, and the axils were reddish. Thus, rather than affecting just one characteristic, the flower color genetically, the gene was actually affecting three traits. Genes like this, which control multiple seemingly unrelated features, are said to be pleiotropic, where pleo means many and tropic means effect. We now know that Mendel's flower color gene specifies a protein that causes colored particles or the pigments to be made. This protein works in several different parts of the pea plant. It's active in the flowers, in the seed coat, and also in the leaf axils. In this way, the seemingly unrelated phenotype can be traced back to detect in one gene, and therefore, if the gene is affected, it's going to affect a lot of different jobs. Importantly, the alleles of pleiotropic genes are transmitted in the same way as the alleles of the genes that affect single trait. Although the phenotype has multiple elements, these elements are specified as packages, and the dominant and recessive version of the packages would appear in the offsprings of the heterozygotes in a ratio of 3 is to 13 is to 13 is to 1, which is the colon. Genes affected in human genetic disorders are often pleiotropic. For example, people with a hereditary disorder called Marfan syndrome may have a set of seemingly unrelated symptoms, including, for example, they usually have a tall height, they have thin fingers and toes, and they have dislocation of the lens of their eye. Heart problems in which the aorta, which is the large blood vessel carrying blood away from the heart, it bulges or it also even ruptures. These symptoms don't seem directly related, but it turns out that they can all be traced back to the mutation of a single gene. This gene encodes a protein that assembles into chains, making elastic fibrils that give strength and flexibility to the body's connective tissue. Mutations that cause Marfan syndrome reduce the amount of functional protein made by the body, which results into less number of fibrils. Now, how does the identity of this gene explain the range of the symptoms? Our eyes and the aortas normally contain many fibrils that maintain structure. That is why these two organs are affected in Marfan syndrome. In addition, the fibrils serve as a storage shelves for growth factors. When there are less number of those fibrils, in case of the Marfan syndrome, for example, the growth factors cannot be shelved and thus it leads to excessive growth. That's why those individuals are really tall and thin. Another example of pleiotropic gene in case of human is variegate porphyria. It is an inherited disorder that is caused by a mutation in the PPOX gene that leads to the buildup of compounds normally involved in the body's production of heme. Heme is an important part of hemoglobin, the protein in the blood that carries the oxygen throughout our bodies. It is used in all body organs. People with this disease have abnormal production of heme. They are very sensitive to the sun whenever they are exposed. That's why they are called photosensitive. And they develop skin blisters and sores when they are exposed to sunlight. People with this disease can also have neurological symptoms in the form of episodes. For example, they can have an acute attack of severe pain in the stomach, nausea, or even vomiting. Symptoms usually begin in adulthood. Varicate porphyria is caused by mutation in the PPOX gene, as I told you, and it is inherited in an autosomal dominant pattern, as you can see on the left side of this slide. Some people who have PPOX gene mutation never have the symptoms of this disease. People with varicate porphyria need to avoid the sun exposure. The attacks that they experience are treated with medication and even hospitalization sometimes. These attacks can also be prevented by avoiding the factors that cause these symptoms. As I told you that varicate porphyria is caused by a mutation in the PPOX gene. Now this PPOX gene is responsible for making an enzyme that called as protoporphyrinogen oxidase. Mutations in PPOX reduce the activity of this enzyme and this allows compounds called porphyrin precursors to build up in the body. These compounds are formed during the normal process of heme production, but they are actually have a reduced activity due to this oxidase, which allows them to accumulate to a toxic level. The acute attacks seen in this disease cannot be brought on by a number of different factors, including certain medications, hormonal changes in the body, alcohol, dieting, and other factors that have not been identified clearly yet. As I told you, it is an inherited disease and it's also autosomal disorder, autosomal dominant. It is transmitted in this manner, and it also means that only one copy of PPOX gene is in each cell, which is actually mutated. All the individual inherit two copies of these genes. Now, in autosomal dominant conditions, as you know, an individual has a disease causing mutation in only one copy of the gene that causes the person to have this disease. The mutation can be inherited from either of the parents, which means it is not sex linked. There is nothing either parent can do before or during the pregnancy to cause a child to have this condition. Each child of an individual with the varicate porphyria has a about 50% or 1 out of 2 chance of inheriting this mutation. Offsprings, unfortunately, who inherit this mutation may be more or less severely affected than their parents. This single mutation reduces the activity of the enzyme, which is, as I told you, required for heme production, and it increases the risk of developing signs and symptoms of porphyria. You have to remember that not all the people who inherit this gene mutation will develop the symptoms of variegate porphyria at any point of their life.
Let's look at another variated form of porphyria, which is called acute intermittent porphyria. And it's actually because of the same type of gene, but it has different phenotypic effects. So let's look at the video explaining that.
Intermittent porphyria is a rare autosomal dominant disorder that belongs to a family of disorders called porphyria. These disorders all affect the production of heme, which is a major component of red blood cells. Now, heme synthesis is also called porphyrin synthesis, and when halted, it results in the buildup of one of its precursor molecules. In order to better understand acute intermittent porphyria, we need to first take a look at hemoglobin. the main protein within red blood cells that's responsible for carrying oxygen. Now, hemoglobin is made up of hemes and globins. There are 4 globin subunits, typically 2 alpha and 2 beta, and each one has its own heme group. This heme group is a large molecule that's made up of 4 pyrrole subunits that forms a ring, and this structure is called a porphyrin. In the middle, there's an ionically bonded iron, and the iron is what binds to and carries the oxygen molecule. So each hemoglobin can carry 4 oxygen molecules when it's fully saturated. Porphyria disorders occur when one of the enzymes in the heme synthesis pathway is deficient, which causes a decrease in heme synthesis and a buildup of metabolites formed in the earlier steps of the pathway. Now, individuals with acute intermittent porphyria have a mutation of the HMBS gene, which codes for the enzyme porphobilinogen deaminase. Without this enzyme, porphobilinogen cannot be converted to hydroxymethylbilane, and the heme synthesis pathway cannot continue. Furthermore, it causes the buildup of the earlier metabolites like porphobilinogen and aminolevulinic acid, which can be toxic to the body. The majority of individuals with the HMBS gene mutation and a deficiency of porphobilinogen deaminase are asymptomatic. Only around 10% of individuals, typically young adult women, are affected by what's known as acute attacks, which are caused by certain triggers. Anything that stimulates increased heme production can be considered a trigger, since it'll inevitably cause buildup of porphobilinogen and aminolevulinic acid. Some examples of these triggers include excessive alcohol consumption, starvation, and certain medications that increase cytochrome P450 protein. This protein also contains heme, and it's used to break down many medications like barbiturates, antiepileptics, and oral contraceptives.
Then we have this phenomena of genetic heterogeneity. Genetic heterogeneity occurs through the production of a single or similar phenotype through specific gene mechanisms. Now, there are 2 types of genetic heterogeneity. We can have allelic heterogeneity and we can also have locus heterogeneity. In case of allelic heterogeneity, it occurs when a similar phenotype is produced by different alleles within the same gene. While in case of locus heterogeneity, it occurs when a similar phenotype is produced by mutations at different loci or locus. Marked genetic heterogeneity is correlated to multiple level of causation in many common human diseases. For example, we have this disease of cystic fibrosis, Alzheimer's disease, autism disorders, and even in the case of inherited predisposition to breast cancer and non-syndromic hearing loss. Now, these levels of causation are complex and they occur either through rare individual mutation that, when combined, contribute to the development of common diseases, or through the accumulation of many different rare individual mutations within the same gene that contribute to the development of same common disease with different individuals. This can also occur through the accumulation of many different rare individual mutations within the same gene that contributes to the development of different phenotypic variation of same common diseases with different individuals. And in the last, the development of the same common disease in different individuals through different mutations. Increased understanding of our role of genetic heterogeneity and the mechanism through which it produces common diseases phenotypes will facilitate the development of effective prevention and treatment methods for these diseases.
So first of all, look at what is cystic fibrosis, and then I'm going to explain to you the genetic heterogeneity behind this disease.
You're probably aware that cystic fibrosis, or CF, is a genetic disorder that affects the lungs. But that's only part of the story. In fact, the name cystic fibrosis refers to the disease's effects on the pancreas, where it can lead to cysts, which are fluid-filled sacs wrapped in a membrane, and fibrosis, excess deposition of connective tissue that can replace or infiltrate normal tissue in an organ. CF is an autosomal recessive disorder involving the CFTR gene, which stands for cystic fibrosis transmembrane conductance regulator. And this gene codes for the CFTR protein. CF develops when there's a mutation in the CFTR gene, but because it's autosomal recessive, you need to inherit 2 mutated CFTR genes, one from mom and one from dad. Now, if mom and dad both have 1 copy of the mutated gene and 1 normal gene, they're considered carriers and they don't have the disease. And inheriting CF is much more common in people of European descent. Now, the CFTR protein is a channel protein that pumps chloride ions into various secretions. Those chloride ions help draw water into the secretions, which ends up thinning them out. The most common mutation with CFTR is the delta F508 mutation. Delta means a deletion, and the F, which can also be written as Phi, is short for phenylalanine, and the 508 is the 508th amino acid in the CFTR protein. So the delta F508 mutation is where the 508th amino acid out of 1480, phenylalanine, is deleted and missing. This CFTR protein with the delta F508 mutation gets misfolded and can't migrate from the endoplasmic reticulum to the cell membrane, meaning there's a lack of CFTR protein on the epithelial surface. And this means that it can't pump chloride ions out, which means water doesn't get drawn in and the secretions are left overly thick.
Okay, so from this video, we came to know that cystic fibrosis is an inherited autosomal recessive genetic disorder that occurs through a mutation in a single gene that codes for the CFTR, and the CFTR gene codes for actually a ABC transporter class, which is an ion channel protein, and that conducts chloride ions across the epithelial cell membrane. Now, mutation of the CFTR gene, which is actually affecting the chloride ion channel function, leads to dysregulation of the epithelial fluids in the lungs, as you have seen in the previous video, and not only in the lungs but also in pancreas and other organs as well, leading to this disease conditions. And you also came to know that the complications include thickened mucus in the lungs with the frequent respiratory infections and pancreatic insufficiency, giving rise to malnutrition and diabetes. Now, these conditions lead to chronic disability and reduced life expectancy as well. Now, research has identified that there are more than 2,000 cystic fibrosis associated mutations in the CFTR gene at varying degree of frequency within the disease carrying population. These mutations also produce varying degree of disease phenotypes and may also work in combination to produce additive phenotypic effects.
Now here we have an example of locus heterogeneity, and we have this example of human disorder called Alzheimer's disease. And you all know that it's a complicated neurodegenerative disorder with multiple phenotypic subtypes. Alzheimer's disease, which is sometimes also referred to as the AD disease or just Alzheimer's, it's a chronic neurodegenerative disease that usually starts slowly and gradually and it worsens over time. It is the cause of 60 to 70% of cases of dementia. The most common early symptom is difficulty in remembering recent event. As the disease advances, symptoms can include problem with language, disorientation, which can be like easily getting lost, mood swings, and loss of motivation as well. And these guys are not able to manage their self-care, and they also have behavioral issues. As a person conditions decline, they often withdraw from family and society. Gradually, body functions are lost, ultimately leading to death. Although the speed of progression can vary, the typical life expectancy following diagnosis is 3 to 9 years. Now, current research has identified a rare mutation in three genes that encode the amyloid precursor protein. This amyloid precursor protein is playing actually a very important role in this disease. And this mutation causes the autosomal dominant, which is actually autosomal dominant disease. It causes an early onset of the familial Alzheimer disease. Research has also discovered the association of a fourth allele, which is apolipoprotein E4, in the development of the late onset and sporadic forms of the disease, although the pathology of its role is still largely unknown. Now we have phenocopy, which is the last thing that we are going to discuss in this series of lectures related to the extensions and modifications of Mendel laws. So what is phenocopy? A phenocopy is a variation in the phenotype, which is the physical appearance, you all know that, which are generally referred to as a single trait, and it is caused by environmental conditions, often but not like necessarily during the organism's development. Now, such that the organism's phenotype matches a phenotype that is determined by a genetic factor. It is not a type of mutation. You have to remember that. And it is a non-heritable disease or a condition. It cannot be transferred from one generation to the next generation. We have several examples of phenocopy. First of all, we have this example of Vanessa butterfly. The butterfly genus Vanessa, it has this ability to change its phenotype based on the local temperature. These butterflies normally migrate from one place to the other place, like they would migrate from European areas such as Lapland to Syria. And when they move from the Lapland, if they are in the Lapland where the temperatures are really cold, they look exactly like the local flies. But if they are localized in Syria, they mimic butterflies of this area. Then we have another example of phenocopy in case of Drosophila melanogaster. Now, it has been found that the larvae of Drosophila melanogaster have been found to be particularly vulnerable to environmental factors which produce phenocopies of known mutations. Now, these factors include temperature, shock, radiation, and various chemical compounds. In fruit fly Drosophila melanogaster, the normal body color is brownish gray with black margins. A heredity mutations for this was actually discovered by T.H. Morgan in 1910, where the body color turned yellow. This was a genotypic character which was constant in both the flies and also in the environment. However, in 1939, it was discovered that if the larvae of the normal flies, which don't have mutation in any of their gene, they were fed with silver salts, they would develop yellow bodied flies irrespective of their genotype. As you can see in the figure in the middle, we have a wild type fly which has this brown body color, and then we have this fly which developed from the larva of that was actually fed with the silver salt. And you see it's all yellow, although it doesn't have mutation in the gene which is responsible for the color of the body. Now, the yellow bodied flies, they are genetically brown in a variant form of the original yellow bodied fly. Then we have another example of Himalayan rabbit. And this Himalayan rabbit you see, both of them are Himalayan, but as I told you in the previous series of lecture, that the Himalayan rabbits are pure white in color, but their ear, their extremities, like their nose and their hands and feet, they are dark colored. Now, the phenocopy, which is observed in the case of Himalayan rabbit, is that when these rabbits are raised in a moderate temperature, the Himalayan rabbits are white in color with a black tail, nose, and ears, which make them phenotypically distinguishable from genetically black rabbits, which are normally present in America, which are called the black rabbits. Now, however, when these Himalayan rabbits, when they are raised in cold temperature, these rabbits are going to show a black coloration of their entire body, which is actually resembling the black rabbits, although they are genetically very different from the black rabbits of America, but they phenotypically they look like them. Now, therefore, we can say that Himalayan rabbits is a phenocopy of the genetically black rabbits. Reversible or a cosmetic modification, such as that of hair bleach, are not considered to be phenocopy as they are not like inherent traits.
So, the main key takeaways from today's lecture are: first of all, we have pleiotropy. This is the condition when one gene controls several functions or has a multiple phenotypes, and that's a mutation in actually one gene that is affecting many aspects of growth and development. Then we have genetic heterogeneity, in which different alleles or genes produce identical phenotypes, and it has two types. We have allelic and we have locus heterogeneity. And finally, we discussed phenocopy. And as I explained you, phenocopy are the environmentally induced traits that resemble a similar phenotype produced by a gene mutation or a genocopy. So, at the end of our series of lecture on the extension and modification of Mendel's laws, here is a brief overview of whatever that we have studied. So in the first column, you see the degree of dominance. Then in the second, you have the description. And in the third one, you have a simple example of what type of dominance that you are actually studying. So first of all, we have complete dominance in which one allele is completely dominant over the other one. And all the traits that Mendel observed, like the color of the seed or the shape of the seed, they are good example of complete dominance. Then we have incomplete dominance, and in that you know that the heterozygous phenotype is between those of the two homozygotes, like the pink flower color, which was produced by crossing a red flower with a white one. Then we have codominance, and in this situation, the heterozygous phenotype results from the expression of both alleles, like we discussed the example of MN blood type in case of humans. Then we have multiple alleles in which more than two alleles for a certain gene or trait are present. And a good example that we studied was about the ABO blood type. Then we have the lethal alleles. These are the alleles which are produced by the mutation in an essential gene, and the time of death is actually dependent upon the gene product that is essential to a developmental stage. When that developmental stage is reached, then it leads to death of the individual. Then we started the example of penetrance and expressivity, in which we started the example of polydactyly. And penetrance is actually the percentage of the individuals that show at least some degree of expression of a mutant genotype, while expressivity is actually the range of expression of the mutant phenotype. We also discussed epistasis, and epistasis is the effect of one gene or a locus which masks or modifies the effect of another gene pair which may be present on another locus. And we learned about that with an example of the coat color in Labrador dogs and the color of summer squash. Then, in today's lecture, we learned about pleiotropy, in which one gene controls several functions and or it has many effects or multiple phenotypes that are produced by that. And we learned the example of porphyria in that. We then learned about genetic heterogeneity, in which different alleles or genes produce an identical phenotype. And the examples were cystic fibrosis and Alzheimer's. Then, in the last, we learned about phenocopy, and the phenocopy is actually environmentally induced trait that resemble a similar phenotype produced by a gene mutation. And we learned about that with the help of examples of Vanessa butterfly and Himalayan rabbit.
Okay, since today we have completed our series of lectures on the extensions and modification of Mendelian laws, here is your assignment. This assignment is actually from the 3 video lectures that we have shared under the heading of the extensions and modifications of Mendelian laws. Now, let me tell you what you have to do with this assignment. First of all, what this assignment is about, and how do you have to do it, and when do you need to submit it. So, what it is about. In this assignment, you will identify different degrees of dominance. You see a table here which has 2 columns, and on the left side of the this table you see that there are different degrees of dominance, like codominance, complete dominance, and incomplete dominance, multiple alleles, pleiotropy, and epistasis. And on the right side you have different examples of that. So what you're going to do is that first you have to match these columns. You have to match the correct degree of dominance with the correct example. And then you have to provide a brief description of the match that you have made. Now the description must be brief and logical. And then the second question is that when you have to submit this assignment, well, you have 2 days to submit this assignment. 2 days after it appears as an assignment on MS Teams or on CU Online. You have to write this assignment on a Word document. You have to then solve it and provide a brief explanation for every match. And then you are going to save it as a PDF file before sending it to me. So, good luck.